Class 9. Last day I have taken a class on dentition. I hope you have gone through the part of the chapter and there are no such problems which you are facing. Now, today I am starting with the first page of the chapter, that is digestive system. As you know, this is also a very important chapter and it is there in your reduced syllabus. Now, first of all, we will go through the definition of digestion. As you can see in your book, they have given two definitions. One is in a short form and another one is a bigger one. Any one of them you can learn. It is the breakdown of naturally occurring foodstuffs into diffusible form, or it is any change which makes the food soluble and of such chemical nature that they can be absorbed through living membranes. Actually, the second definition is more appropriate, but it's upon you which one you want to learn. Now, in the digestive system, a very big role is played by the enzymes. I hope you all know what are enzymes and how does they work, but here we will know a bit elaborately about them. Now, enzymes can be of any nature, like it can be protein, it can be steroid, it can be amino acids, peptides, but they are actually biocatalysts. Now, I hope you all know what are catalysts. Now, catalyst means those which alters the rate of the reaction but themselves doesn't take part inside the reaction, right? Now, enzymes also work in the same way, but because it is required for our physiological processes, so we call it as biocatalyst. Now, what are the characteristics of an enzyme? They have given 7. At least 5 to 6 you have to learn. It's a protein and is therefore destroyed by heating. It acts only on one kind of substance called the substrate, that is, it is specific. Now, you must remember that enzymes are specifically protein in nature. Other kinds of enzymes are there, but primarily they are all proteins or peptides or amino acids, right? It acts only on one kind of substance called the substrate, that is, it is specific. It always forms the same end product from the substrate. It only affects the rate of a chemical reaction and always speeds up the reaction. Like a catalyst, it can be used again and again. It acts best only at a particular pH and it acts best within a narrow temperature range, usually between 35 to 40 degrees centigrade, and we generally call this temperature as the optimum temperature. Now, digestive system consists of two main parts, alimentary canal and digestive glands. Now, it's a muscular tube, the alimentary canal, which starts with the mouth and ends at the anus. And they have got various digestive glands, liver, pancreas, salivary glands, right? Now, we'll start with the alimentary canal portion and we'll start from the mouth. It is a space where the food is chewed and mixed with saliva. Its front limits are formed by the upper and lower lips. The lips help in closing the mouth, sucking and sipping liquids, speaking, and also in perceiving certain sensations like touch and heat. Now, inside our mouth, we all have a tongue, and we know that without tongue, even we cannot speak. Other than that, it also consists of the taste buds. So, tongue is a muscular part of our mouth, which helps in manipulating the food while chewing and mixing it with the saliva, for tasting, for cleaning the food particles from the teeth, and also in speaking. So these functions are very important. Now, dentition we have already done. So again, I'll shift to the next part of the chapter. The first gland is the salivary gland. Now, salivary gland consists of 3 pairs of its parts. One is parotid glands, another is submandibular, another is sublingual. Now, students, you must know their proper location. Parotid glands located just in front of and beneath each ear. Submandibular lying close to the inner side of the lower jaw on each side. Sublingual below the tongue. So, their locations are very, very important. Now, saliva is very slightly acidic, that is pH 6.8. As you know, 7 is the neutral one. Fluid containing water, about 99%. salts, mucus, and an enzyme, salivary amylase. We also call the salivary amylase as talin. Any one of you, any one of them you can write in your exam, and also you may get question with any one of the names. So both the names you must remember. Now, what are the functions of this saliva? Saliva is very important for us to digest the food. First of all, it moistens and lubricates the inner lining of the mouth cavity. It's a printing mistake. It's written moral cavity, it should be mouth cavity. And the surface of the tongue to facilitate speaking and swallowing. It moistens and lubricates food, which again helps in swallowing. Acts as a solvent, dissolving some food particles to stimulate taste buds of the tongue. Helps food particles to stick together to form bolus so that they can be swallowed in a mass. Another very important function of saliva is the reaction in which it is helping. That is, it digests starch. Its enzyme talin converts starch into maltose. This is a very important reaction because the digestive reactions are starting from here only, and this explains why if boiled rice is stewed very well, it begins to taste sweet. So see the reaction: starch with the help of salivary amylase, it's being converted to maltose. Cleans the mouth and tends to destroy germs to prevent tooth decay. And dryness in mouth gives a feeling of thirst to replenish food body water. Thus, saliva aids in water balance in the body. Right. Now, they have given so many functions. At least any 5 you have to learn, and also you have to practice the reaction part. Okay. Now, swallowing and peristalsis, the tongue presses upward and back against the roof. We call it as palate of the mouth, and forces the bolus. Bolus means the ball of the chewed food into the throat or the pharynx. The back part of the roof of the mouth cavity, we call it as soft palate, closes the opening between the throat and the nasal passage. Larynx, you know larynx, it is a voice box with which the voice is coming out while we are speaking, is located at the entrance of the windpipe, is pulled upward to bring it close to the back of the tongue when a flap called epiglottis closes its opening. Now students, this epiglottis is very important because we must know where is it located and what is its function actually. It's a flap which helps the food to go inside the food pipe rather than to the air pipe. Right. Once the food reaches the esophagus, it is conducted behind by a special movement called peristalsis. Now we must learn the definition of peristalsis. It is the wave of constrictions caused by the circular muscles of the gut pushing the food along. This definition is very, very important. As the wave passes, the circular muscles relax, and peristalsis occurs where? Throughout the regions of the gut. Gut means the alimentary canal. Right. Now, next part of the digestive system is the stomach. It is an elastic bag located below the diaphragm. In an average adult, it can hold 2 to 3 liters of food. Its walls are highly muscular and churn the food thoroughly to mix with the gastric juice secreted by the inner lining. Now, the digestive juices which are coming out from the stomach, we call it as gastric juice. The opening of the stomach into the intestine is called pylorus. Very, very important for your short questions. It has a ring of muscles known as sphincter muscles to keep the opening closed like a valve to prevent food passing from the stomach until it is thoroughly churned up. Now, gastric juice is secreted by the inner lining of the stomach. It is a colorless, highly acidic liquid containing water, salts, hydrochloric acid, and an enzyme which is very important called pepsin. Now, what is the function of this hydrochloric acid? It serves 2 functions. You will have to remember it. It kills any germs which may have entered along with the food. And number 2, it activates pepsin to act on proteins. Now, how is it activating pepsin? That we have to go through the reaction. In fact, the pepsin is first secreted as pepsinogen, which is then changed to pepsin by the help of this acid. Pepsin digests proteins into peptides. Now see the reactions. Very important, you have to practice them a lot. Pepsinogen is the inactive form. HCl is acting and converting it to pepsin. Now, proteins are being acted upon by the pepsin and they are being transferred to the peptides. Similarly, prorenin by HCl forming renin. Now, this renin is the enzyme which is acting on the milk protein or casein in case of babies who all have milk. Okay, and it is changed into paracasein, which is an insoluble form of the milk protein. Right. So, these are the four reactions we must know. And what is chyme? That also you have to learn. The food stays in the stomach for about 3 hours and it attains a pulp-like form called the chyme. Now, next part of the digestive system, the small intestine. The small intestine is a tube about 7 meters long. It is coiled and folded in the abdomen. There are three subregions, duodenum, jejunum, and ileum. Now, the inner lining of the ileum is produced into a great number of tiny finger-like projections called villi. Now, students, the diagram of villi is very, very important. This one. Figure 11.6 in my book. With proper labeling. Very important diagram. You have to practice it. Okay. Now, the villi enormously increases the inner surface area of the intestines, which facilitates the absorption of digested food. Between the villi are small holes through which the intestinal juice secreted by its glandular cells is poured into the lumen of the intestine. Each villus is covered by a single cell thick epithelium. Inside the villus are contained an artery, a vein, interconnecting blood capillaries, and a lymph vessel called lacteal. Very, very important. What are the parts present inside the villus? Villus is the singular form and villi is the plural. The small intestine serves both for digestion and absorption. There are two digestive juices. One is bile, I hope you all know about it, and another one is pancreatic juice. Now, what are the adaptations of the ileum for absorption of digested food? They have given 4 points. All the 4 points you have to learn. First of all, it is very long, so definitely more surface area. The large number of villi further increases the surface area. Single cell epithelium, easy for penetration. It is narrow for slow movement of food, allowing absorption. Okay, so all the 4 points are important. Now, next we'll come to the bile. Now, this diagram is also very important, 11.7. The bile, it's a yellowish green watery fluid produced in the liver, which is transported through the hepatic duct. It is joined by the cystic duct to form the common bile duct. This line is very important. Okay. Now, the color of the bile is due to certain pigments. Now, both the names of the pigments are very important. Biliverdin and bilirubin, produced by the breakdown of the dead and worn out red blood cells. This also you have to remember from where we are getting these two pigments. Okay. It contains a lot of sodium bicarbonate which neutralizes the acid content of the food received from the stomach and makes it alkaline to enable the pancreatic and intestinal enzymes to act. Bile salts reduce the surface tension of fats and break them into tiny droplets. We call this process as emulsification. See the two reactions. Fat with the help of bile forming emulsified fat. Another one is acidic chyme with the help of bile and sodium bicarbonate forming alkaline chyme. Similarly, another digestive juice is the pancreatic juice. They have got three kinds of enzymes. Amylopsin, another name is pancreatic amylase, trypsin and stepsin. Again, as we have seen in case of pepsin previously, like that only, trypsin also, it is first secreted in the form of an inactive trypsinogen which is activated to trypsin by an enzyme known as enterokinase. So first of all, we'll go to the reaction and we'll see that trypsinogen is converted to trypsin with the help of enterokinase. Now the proteins and peptides are being acted upon by trypsin to form smaller peptides and amino acids. Leftover starch are acted upon by the amylase, that is amylopsin actually, forming maltose, and emulsified fat are acted upon by the stepsin, forming fatty acids plus glycerol. Okay. Now, intestinal juice contains a lot of enzymes. Erepsin, known as peptidases, that is acting on peptides and converting them to amino acids. Similarly, maltase acting on maltose, forming glucose. Again, sucrase acting on sucrose, forming glucose plus fructose. Lactase acting on lactose, forming glucose plus galactose. Lipase acting on emulsified fat, forming fatty acids and glycerol. So, students, first of all, this is very easy to remember because there are a lot of names which you have to remember definitely, and for that, you have to practice this by writing a lot of time. Other than that, it's very important to remember these names in the form of like the substrate on which they are acting. Actually, their names are derived from there only, like maltose, maltase, sucrose, sucrase, lactose, lactase. Like that, it's very easy to remember, right? Now, absorption of food, the final products, the amino acids and simple sugars have all relatively small sized molecules which are absorbed through the thin epithelium of the villi and reach their blood capillaries to finally enter the blood circulation. The fatty acids and glycerol are absorbed into the lymph vessels or the lacteals, we have just read about it, to enter the lymphatic system, which forms a network all over the body to ultimately empty its contents into the bloodstream. The large intestine. Till here we have read so many things about small intestine. Now it's the large intestine. It is about 1.5 meters long. It has three parts: cecum, colon and rectum. Now cecum is a small blind pouch situated at the junction of the small and large intestine. So first of all, location is very important. And another thing which is very important over here is the vermiform appendix. You know, appendix, I have heard about appendicitis. It's the inflammation of this appendix. And today it is a vestigial organ. It's a functionless organ. Primitively it was important to us, but now it is a functionless organ. The colon is much broader than the ileum and is a little more than a meter long. And rectum is the last part, about 15 centimeter long, which opens at the anus. The anus has circular muscles called sphincter muscles to keep it closed except when passing bowels. Now you must remember that wherever we are talking about the circular muscles, I think you have noticed it, that everywhere we are talking about the sphincters. Actually, the circular muscles, wherever is it located, it is known as sphincter muscle. Clear? This part is not so important. Another very important definition is assimilation, because after absorption we need to assimilate the things into our blood. So assimilation is the conversion of the absorbed digested food into body material. Okay. Now the liver converts any excess glucose into insoluble glycogen, which can be temporarily stored. The amino acids circulate in the body and they serve as building blocks of protein. Any excess amino acids are broken down in the liver by a process called deamination. The fatty acids and glycerol absorbed by the gut are transported mainly through the intestinal villi and lymphatic system. So the parts only through which I have gone through from here, that much only is required. Okay. Now, liver is the largest gland of the body, weighing about 1500 gram, reddish brown in color, located in the upper right side of the abdomen, just below the diaphragm. This much is very important. Now, this part is not in syllabus, the functions of liver, so it will not come in your exam. Now, students, they have given some of the experiments on digestion. Please go through the experiments because this part is there in your practical. Okay. So I will definitely describe this when I will go through the practical. This part you just go through once. See, this part also. This part you just go through once. Okay. Otherwise, we are done with the chapter. Clear? Now, please go through the chapter properly. If there are any problems, you can come up with that in your comment box. And please, there are very few diagrams in this chapter, but there are a lot of things to remember, especially the reactions. Jot it down and practice it. Okay? Thank you.